Today, we're diving into a Nobel Prize-winning breakthrough that's so fundamental, it's like we've finally found the Rosetta Stone for biology itself. We are talking about technology that is not just unlocking, but is now starting to rewrite the very code of life.
All right, so here's our game plan. We're going to start with a 50-year-old puzzle that had scientists completely stumped. Then we'll see how a revolutionary AI finally cracked the code and explore this mind-blowing shift from just predicting life to actually designing it. After that, we'll look at some of the incredible applications, pull back for a necessary reality check, and finally, think about what it all means to engineer our future.
To really get why this is such a huge deal, we have to rewind the clock a bit and understand this monumental challenge that was sitting right at the heart of biochemistry for half a century.
It all starts with these amazing little things called proteins. I mean, they are literally the workhorses of every living thing on the planet. Think of them as these brilliant microscopic machines that do pretty much everything. Building our muscles, fighting off viruses, you name it. And all of their power comes from their incredibly complex, unique 3D shapes.
And this, right here, was the massive roadblock. For decades, scientists could easily figure out the sequence of amino acids, the string of beads that make up a protein. That was the easy part. But predicting how that simple, one-dimensional string would twist and fold itself into a functional three-dimensional machine, that was one of the hardest problems in all of science. It was like knowing all the letters in a sentence, but having absolutely no idea what the words mean.
And look, this wasn't some abstract problem for scientists in a lab. It had huge real-world consequences. Just take a look at how we used to discover drugs. It's this long, incredibly expensive, and mostly unsuccessful process. And why? Well, a big reason is that without knowing a protein's exact shape, trying to find a drug molecule that fits it perfectly was like fumbling around in the dark, trying to design a key for a lock you've never even seen.
So, how do you solve a problem that's been stumping the smartest minds for 50 years? Well, it turns out you don't just tweak the old methods. Nope, you bring in a totally new player to the game, a revolutionary AI that was about to change biology forever.
So, the 2024 Nobel Prize in Chemistry went to these three absolute pioneers for their connected breakthroughs. Demis Hassabis and John Jumper from Google DeepMind were honored for their incredible work on protein structure prediction. And David Baker from the University of Washington was recognized for his groundbreaking work in both prediction and what comes next, protein design. I mean, just look at this timeline. It tells the whole story. For more than 60 years, progress is just slow and steady, painstaking work. Then, boom, in 2018, the first version of AlphaFold shows up to this competition. It's kind of like the Olympics for protein prediction, and it just blows everyone away. A few years later, AlphaFold 2 comes out and basically solves the problem. It was a complete quantum leap.
And when I say quantum leap, I am not kidding. The sheer scale of this is just mind-boggling. Before this, the public database of protein structures, which took scientists 50 years to build, had about 170,000 entries. AlphaFold predicted the structures of over 200 million proteins, a 1,000-fold increase, and then they just gave all that data away, for free, to every single scientist on the planet.
And of course, they didn't just stop there. The newest version, AlphaFold 3, goes even further. Now it's not just about predicting the shape of one protein in isolation. It can predict how that protein will interact with almost everything else inside a cell, with drugs, with DNA, with RNA. It's giving us a picture of how all the machinery of life actually works together.
Okay, so being able to predict the shape of any protein nature has ever made, that's incredible. But what if you could go beyond that? What if you could dream up entirely new proteins that have never existed before and build them from scratch?
Nobel laureate David Baker put it perfectly with this amazing analogy. For a long time, if you wanted to change a protein, you'd basically just tinker with one that nature already made. You'd modify the bird. Baker's whole idea was different. He wanted to understand the fundamental rules, the aerodynamics of proteins, so we could build completely new flying machines. And that's the whole concept of de novo design, which just means from the new. You don't start with an existing protein. Instead, you sit down at a computer and say, okay, I need a molecular machine that does this specific job and looks like this. Then you use the basic laws of physics and chemistry to figure out the exact amino acid sequence that will fold itself into that shape you just designed.
This was the watershed moment, years before AlphaFold was even a thing. Baker's lab pulled off this incredible proof of concept. They designed a protein called Top7 on a computer, a shape never seen in nature. Then they made it in the lab, and guess what?
The real-life structure was a near-perfect match for their computer design. This was the aha moment. It proved that yes, we really do understand the rules, and now we can be architects at the molecular level.
So this is the absolute key difference you need to get. Think of it this way. AlphaFold, the prediction tool, is like an expert translator. It can read the book of life that's already been written. But de novo design, that lets us become authors. It lets us write brand new chapters for the book of life. Okay, so we can predict, we can design. So what? What can we actually do with this power? This is where the science goes from being a really cool idea to something that can change the world.
Well, right out of the gate, the impact on medicine is just staggering. We're talking about designing drugs that are so precise they can target just cancer cells and leave healthy cells alone. We can speed up drug discovery like never before, and we can create custom-built molecules to fight drug resistance in viruses like HIV and coronaviruses.
And it's not just treatments, it's prevention too. Think about vaccines. Imagine designing a vaccine that shows your immune system the most vulnerable part of a virus, creating a super powerful and precise response. Or being able to design a new vaccine almost overnight when a virus mutates. That's the future this technology is building.
And look, the potential here goes way beyond just us. We're talking about tackling some of the planet's biggest problems. Scientists are already designing custom enzymes that can literally eat plastic, breaking it down, or designing proteins that can pull carbon dioxide right out of the air. The possibilities are enormous.
All right, with all this amazing, world-changing potential, it is really easy to let the hype run away. So we need to hit the pause button for a second and talk about a little reality check.
So, is this AI the magic bullet, the perfect solution to everything? The short answer is no, not yet anyway. And it's really important to understand why, because knowing the limits of a tool is what lets you use it wisely.
Here are some of the biggest hurdles they're still working on. First, proteins aren't statues. They're constantly moving, wiggling, and changing shape. AlphaFold gives us an amazing snapshot, but it doesn't give us the movie. The models can also just get the shape wrong sometimes, especially for really flexible proteins. And the accuracy isn't as good for orphan proteins, ones that have no known evolutionary relatives. And then there's this really weird quirk called the hallucination effect.
So what is that? Well, some parts of proteins are just floppy. They're naturally messy and disordered. But the AI is trained to find patterns, to find order. So sometimes it looks at that mess and kind of just makes something up. It invents a structure that isn't really there, which could obviously be a big problem if you're not careful.
And this points to a fundamental challenge with any AI, really. It can only ever be as smart as the data you train it on. AlphaFold learned its stuff from that protein databank we talked about earlier. But that database, as big as it is still incomplete. So the AI's knowledge of the biological universe is also incomplete.
So, we've seen the incredible power, but we also see the very real limitations. So what's the takeaway here? Where do we go from here?
The single most important thing to remember is this. These AI models are not replacing scientists. They are supercharging them. Think of it as a revolutionary new tool in their toolkit, an incredibly powerful one, but still a tool. It helps them form better ideas and design smarter experiments faster than ever before.
This chart shows you exactly what I mean. Look at AlphaFold 3's accuracy in predicting how a drug might connect to a protein. It's a massive improvement over older tools, right? But it's still not 100%. There's still almost a 1 in 4 chance it's wrong. And that gap, that's where the human scientist in the lab is still absolutely essential.
And this quote from the research just nails it. The real magic isn't AI or experiments. It's AI and experiments working together. The AI can point you in the right direction with incredible speed, but it's the real-world experiments that confirm you've actually reached your destination.
And all of this leaves us with this final massive question. For the very first time in human history, we're moving from just being observers of biology to being its authors. We can read the code of life, we can predict it, and now we can start to write it. The scientific question of, can we do this, is, well, it's basically been answered. And now we face a much bigger, much harder question, a question for all of us. What kind of world will we choose to build?